Our transcription adventure begins when an enzyme called helicase travels along a double helix of DNA, and this begins to open it, providing a single strand of DNA, which is to be used for complementary base pairing between the single DNA strand and RNA polymerase. For RNA polymerase binds to a promoter region of the single strand of DNA that contains a specific sequence of nucleotides, for then this begins to create an unprocessed mRNA strand by complementary base pairing as it runs along the strand. But in lieu of the nitrogenous base thymine, the base uracil is used in the complementary pairing with adenine instead of thymine. Once the complementary base pairing is completed, we are left with a complete primary mRNA. But if this mRNA strand is to be used for protein synthesis, it must be processed. Processing. The first step in processing the RNA is to add an addition of a cap to one of the ends of the primary mRNA. This cap is composed of an altered guanine nucleotide, while the other end of the RNA is modified by the addition of a poly-A tail. This is composed of multiple adenosine nucleotides. For it to be processed, we must remove the introns out of the strand, which we have represented by the pink parts in the mRNA strand. For these are segments of DNA that are not part of the gene, which will not be expressed, but for the ones that will be expressed, they are called exons, represented in blue. From there, a complex of multiple enzymes and proteins and RNA, or SNRPs as they are called, surround the introns that are represented by the pom-poms and remove the introns from the strand and bind the remaining exons together to make a continuous strand of mature messenger RNA. For then this mature messenger RNA is ready to leave the nucleus and enter the cytoplasm.
Translation. Initiation. Once the messenger RNA exits the nucleus and enters the cytoplasm, the ribosomal subunits bind to the mRNA and forms a ribosome. The ribosome is composed of two subunits, one large and one small, and is composed of ribosomal RNA. The ribosome then starts to move across the mRNA, and transfer RNA, or tRNA, brings amino acids to the ribosome. Messenger RNA is read 3 bases at a time and always starts with methionine, represented by AUG, and is then matched by the correct anticodon on a tRNA. Elongation. The tRNA arrive in the A site and continually move to the P site, then exit through the E site to exit the ribosome. As the triplets are read, the tRNA's amino acids detach from the tRNA and then forms a polypeptide bond with the adjacent amino acid to form an amino acid chain. Termination. Once the mRNA's stop codon has been read, a tRNA without an amino acid arrives in the A site and signals to stop translation. For then a protein called the release factor binds in the A site and the two ribosomal subunits separate from the mRNA. The amino acid chain then folds into a complex shape to form the protein and is sent to the endoplasmic reticulum.